And now we're happy to welcome Cereno Scientific and CEO Sten and Sörensen. Welcome, Sten.
Thank you. So I'm pleased to be here and to present to you the exciting development that we have with Cereno Scientific over the last period. So Cereno Scientific develops novel drugs to transform treatment for PH and other cardiovascular diseases. And we are working with the vision to uncouple the epigenetic modulation through HDACi inhibition for cardiovascular disease. We have three programs in development: CS1 in phase 2 in PH, performed in the US in the clinical study. And then we have two preclinical programs: CS585, a preclinical prostacyclin receptor agonist, and we have CS014, which is a preclinical HDAC inhibitor, a novel HDAC inhibitor. We have a global presence, headquarters in Gothenburg, US subsidiary Cereno Inc. in Boston, and quite a large research going on in the US with a phase 2 study and two preclinical programs in collaboration with University of Michigan. What we're doing has attracted top leaders in the field to our scientific advisory board. Dr. Bertram Pitt has pursued a survival trial with the mineralocorticoid receptor antagonist on my initiative in the 90s, and Dr. Fayez Sanad was part of that study, but also a survival trial in heart failure with beta blockers when I was at AstraZeneca. And those two trials changed the paradigm for treatment in heart failure with these two agents, and they're used in 95% plus of the patients today. In addition, we have two members from Harvard Medical School, and we have Dr. Gunnar Olsson, the former head of clinical research for cardiovascular at AstraZeneca, and the latest addition, Dr. Raymond Benza, top leader in the PH field in the world. So the three programs: CS1 in phase 2 for PH, top-line data targeted for the end of this year. And then we have the two preclinical programs with a target to have an IND submitted by mid next year and go into phase 1 for each of these two programs. So by next fall, we would have three programs in the clinic in cardiovascular disease. Let's go to our first program, CS1, the HDAC inhibitor for PH. PH is a very severe disease that leads to shortness of breath and eventually a too short life. And it is impacted by morphology changes in the lungs and a buildup of pressure. And eventually the right heart fails and you die unless you get a lung heart transplantation, which very few gets. So there is a massive need for new drugs that can improve the treatment and also life expectancy for these patients. The market is fairly large, approaching $10 billion US, but there is a very large unmet need. All of the drugs that are used today and approved by the FDA are vasodilators. And there are quite some side effects with these, and they're also difficult to take. But the main thing with these drugs is that they lower pressure through the vasodilatation. And the market is looking for something more than that. The same way the heart failure market was looking for new agents back in the 90s and early 2000s. So there is a large unmet need for therapies that can treat the underlying pathophysiology here and have an impact on disease progression and maybe reverse it. Also safer and more tolerable treatments, and the administration would be better. Some of these drugs are taken 8 times through inhalation per day. Here's where CS1 and HDAC inhibition comes in. So it might be game-changing for these patients, and that is due to the way HDAC inhibitors act. And epigenetic modulation, you don't change DNA, you just change the expression of proteins that are mapped in the DNA code. And by doing so, you can help patients with cardiovascular disease. And this is the vision that Dr. Sverker Järn had when he founded the company. The scientific advisory board, when they were brought together, immediately pointed to us that this is a much larger concept than we originally thought. And they pulled together an article that was published last year in the Lancet, which is one of the most prestigious peer-reviewed journals in the scientific field. And this article points out to all the positive documentation that's there in the preclinical and clinical stage. And so there's a lot of things that can be done with HDAC inhibition if it's pursued by the pharma industry and biotech industry. Since that evaluation, publication, and prior to that, we did a strategic review and focused on pulmonary arterial hypertension, the orphan disease, as our first indication. But there are a number of other indications that we can pursue. So it's like a platform for research for cardiovascular disease. The first vision that Professor Sverker has, and our first focus, was actually prevention of thrombosis. And the upgrade of TPA, a fibrinolytic substance, in risk patients to prevent thrombosis. So the body has its own fibrinolytic system that's released over a clot before it becomes occlusive. And in risk patients, TPA is in two small concentrations in the endothelial cell walls. By giving our compound to these patients, we would upgrade the synthesis, storage, and release capacity so the patients would be better prevented by its own defense system. We have documented that we can do that in preclinical studies, but also impact TPA in man in experimental studies. We have also documented that the main blocker of TPA in the body, pi, can be reduced significantly in the morning hours when it's dangerous for these patients. But we have moved on to PH as our key indication. And why is this field hot? I mentioned before that there is a very large unmet need, and it's benchmarked by this deal in November when Merck bought Acceleron for $11.5 billion. And the reason they bought Acceleron was that they have a drug for PH that might be disease modifying, and they're positioning it as a reverse modifying agent. That's exactly the field that we are with our HDAC inhibitors and CS1 in phase 2. And if you look at this slide, we have compared Sotatercept, which was the drug that Merck bought in November, and the capacity that that drug has shown in preclinical data. And as you can see, it's positioned, it has an orphan drug designation, so do we have potential disease modifying? Pulmonary pressure reduction is a given. Reverse remodeling, they claim, and antifibrotic, anti-inflammatory. And so do we through our studies in the preclinic. In addition, CS1 and our substance have proven antithrombotic capacity, which is a manifestation that PH patients suffer at the end of their disease. I won't go into the detail of this quite complicated study. It's more to showcase that these are 4 studies in proof of concept studies in rat in pulmonary arterial hypertension, 2 with our substance, VPA, and 2 with Sotatercept. And they basically show similar kind of data when you compare them. So they both reduce pulmonary pressure, they both have reverse remodeling, they are both antithrombotic, they are both anti-inflammatory, but only then CS1 is antithrombotic. The difference between these 2 drugs is mainly that we are an HDAC inhibitor with CS1, and Sotatercept is a TGF-beta ligand trap molecule. So they have 2 different mode of actions, but it looks very similar in the preclinical work. Now, Sotatercept is in phase 3, and we are in phase 2 with our CS1 compound. If you look at the drugs that are on the market today, as I mentioned, they are vasodilators, and that's how FDA has approved them. They are basically have the efficacy of lowering the pressure, and you improve the 6-minute walk test, so the working capacity of the heart and the lungs. And you can see here HDAC inhibitor, which we have, is not yet on the market, and the drugs below are in development. And the first one you see there is Sotatercept. And as you can see here, we have tried to qualify the efficacy parameters that the different drugs have. And all above HDAC inhibition are vasodilators, and the new drugs are targeting reverse remodeling to have an impact on disease modification. And so do we. And we believe that we are maybe the best drug here, similar to Sotatercept, but a different mode of action. And we are the only drug that has antithrombotic capacity. So these 4 or 5 capacities that we have identified with HDAC inhibition: pulmonary pressure reduction, reverse remodeling, antifibrotic, anti-inflammatory, and antithrombotic, fits like a hand in a glove for this disease. But it also, these various efficacy parameters could have a distinct impact on disease progression in many cardiovascular diseases. We are pursuing PH first. Now let's turn our focus to another proof of concept that we have documented that actually impacts disease progression. This is genetically disposed mice for connective tissue development, so fibrosis in the vessels and in the heart. And you might know that atrial fibrillation is the main cause of stroke, so blood clots in the brain. In man, and that's how all the anticoagulants and antiplatelets are used. And in a way, this model is mimicking that development. So atrial fibrillation is caused by connective tissue development in the heart in man, and then that leads to stroke in the brain. These mice develop connective tissue in their vessels and in their heart. They develop atrial fibrillation and then a thrombosis in the heart in this model. And if you use our substance, VPA, compared to control, you see that there is less connective tissue development, later onset of atrial fibrillation, and less thrombosis. So this, in a way, shows impact on disease progression by our active substance, VPA, in a stroke model or a thrombosis model caused by atrial fibrillation. Very interesting data. This is another proof of concept study where we show that if you have treated mice 96 hours before you hurt the mesenteric veins, so you get a small bleeding and then a clot formation, these mice are better off, so they get less thrombosis if they are pre-treated with our HDAC inhibitor, because they have then the TPA to dissolve the clot before it becomes occlusive. Very interesting data published in 2017. So there is a number of very intriguing preclinical data on HDAC inhibition in cardiovascular disease. We have brought VPA into a unique formulation to fit for drug use in cardiovascular disease, and this phase 1 trial showed a significant reduction of PI in the morning hours, which is very important as PI blocks TPA, your fibrinolytic substance. We didn't see any bleeds of the treatment, of course, and we had the targeted pharmacokinetics that we were after. So we have brought CS1 into a phase 2 trial, and as you can see from this quote from our principal investigator and the key opinion leader, Dr. Raymond Benza, this could be game-changing for these patients. We might be able to not only lower the pressure, increase the work capacity, but also have a reverse remodeling capacity. Now, another very interesting fact here is that we have been able to negotiate a collaboration with Abbott. Abbott has a cutting-edge technology to measure blood pressure in the lungs that's implantable. So we are collaborating with Abbott in our phase 2 trial in PH in the US, where the implantable device is put into the patients to measure the pressure continuously and when we want it measured. This is the first time in Abbott's history of 120 years that they ever done this kind of collaboration, so that makes us both proud and excited. The study in PH is in 10 centers, 30 patients, and after an implantation of the CardioMEMS technology, 6 weeks balancing, and then randomization out to 3 dosage strengths with CS1. And then we wait 12 weeks and we measure a number of parameters. The most important here is safety and tolerance, but we measure all the standard parameters for efficacy that's measured normally in pH, and then some. So for instance, we have as secondary objectives to include a change from baseline on data from CardioMEMS, echocardiography and MRI. And these will be evaluated blinded at the certified course centers. And you can see the various morphology and functional parameters that we are measuring with these various techniques. Let's focus on our preclinical pipeline. So we have 2 programs, and they are way past our 2-year program that we have agreed to run at the University of Michigan in a collaboration program. And we just nominated CD nomination for both of these compounds, the CS585, the prostacyclin agonist, and CS014, which is a novel HDAC inhibitor. We're quite excited about these programs. We aim to bring the prostacyclin agonist for an IND submission by next summer. This compound we have signed an option, first option to acquire in a licensing agreement with University of Michigan for global development and commercialization if we choose to do so after the 2-year program is over. And so far it looks very good. Then we have the HDAC inhibitor. That drug was acquired from Emeriti Bio in 2019, and we just CD nominated that for IND submission by next year, and we will bring that into clinical phase 1 by the fall next year if successfully pursued. Very interestingly, we have released data from our 2 preclinical programs. The first one is the European Hematology Association, EHA. CS585 is a first-in-class compound targeting the IP receptor for prevention of thrombosis without increased risk of bleeding. And at the ESC, CS014 is a novel HDAC inhibitor regulating platelet activity for fibrinolysis and clot stability for prevention of thrombosis without increased risk of bleeding. And then our study in phase 2 has been accepted at PVRI. So by that I will conclude that we are off to a good start in 2022. We have solidified our IP position, we have strengthened our executive management team, and we have nominated our 2 preclinical programs, and they both got accepted at major congresses with new, very exciting data. We are waiting for our first patient to be enrolled in our phase 2 trial, and we have a financing event coming up and a warrant series in September, and then top-line data by the end of the year. By that, I conclude the talk. Thank you.
Thank you, Sten. Like you mentioned here, you now have 3 programs in development. What does this mean for the demand on your internal resources? I'm thinking both in terms of staff and in terms of your finances.
Well, it is. We are very, very busy at the moment, and we have recruited a new head of preclinical development. And earlier this year, we recruited a head of clinical development. So we are strengthening our staff. And we have in our quarterly report that was released, we had 77 million in the bank from the last successful financing, the warrant package T01 in September last year, which was prescribed to 97%. We have another financial events, the T02 package, warrant package in September. And if that's fully prescribed, we'll get another 114 million gross into the company. So we feel we're pretty well financed at the moment, and that the next financing will take us way into our phase 1 programs. So we're fine.
So just to finish off then, you mentioned here you're participating in several conferences. Do you have a set goal with your participation? Yeah, so of course we are here presenting us, and so we have a responsibility to be transparent and communicative to our shareholders, current and new ones perhaps. So we do that very actively, I think. And then we also now, when we are getting more substance and can be more
communicative around our preclinical programs. In addition to our clinical programs, we are submitting that data, and as you saw, it's been accepted. Very interesting data. And that creates some attention around in both the investing and the partnering world. So big pharma industry, I'm sure they're looking out for what Cereno is doing at the moment.
Well, thank you so much for your presentation, Sten.
Thank you. Pleasure.